Hello and welcome everybody to my channel Umini Bio Entrance. Today we will be discussing the NEST 2014 question paper. I will be discussing only the biology. So coming to the first question. Before that, the first section contains 14 questions. For questions 21 to 210, only one of the four options is correct. A correct answer will earn 3 marks and a wrong answer will earn -1 mark, and an unattempted question will earn 0 marks. So coming to the first question. The antigen binding site of the antibody molecule is composed of option A, light chains only, B, heavy chains only, C, the Fc fragment, D, both light and heavy chains. Answer is option D, both light and heavy chains. From the picture you can see that this is the structure of an immunoglobulin. This is the light chain, this is the heavy chain. Both are linked by disulfide bonds, as well as both the heavy chains are also linked by disulfide bonds. Then you have got the constant region of both the heavy chain and the light chain. Then there is the variable region, both heavy and light chain, and this is the antigen binding site. So both light and heavy chains will compose the antigen binding site of the antibody molecule. So answer is option D. Next one. Induced pluripotent cells are formed by isolating and growing cells from the early embryo, fusing enucleated ovum with adult somatic cells, inserting a few relevant genes into adult somatic cells. That is correct. Fusing egg and sperm. For your information, I will tell you. Induced pluripotent stem cells are the type of pluripotent stem cells that can be generated directly from a somatic cell. So before that, what is pluripotent stem cells? Pluripotent stem cells are nothing but those cells that have the capacity to self-renew by dividing and to develop into 3 primary germ cell layers of the early embryo, and therefore into all cells of the adult body. Okay, that is meant by pluripotent stem cells. Now these pluripotent stem cells can be generated directly from somatic cells. This technology, called as induced pluripotent stem cell technology, was pioneered by Shinya Yamanaka, who showed in 2006 that introduction of 4 specific genes known as Yamanaka factors, which encode transcription factors, could convert somatic cells into pluripotent stem cells. Since these induced pluripotent stem cells can be derived directly from adult tissues, that is somatic cells, they bypass the need for embryos. So answer is option. Next one. Evolutionary relationships between species such as humans and apes can be studied by using immunological techniques to compare proteins. A rabbit, when injected with human serum protein albumin, forms antibodies against it. These antibodies are extracted and purified. A fixed amount of antibody is reacted with a fixed amount of albumin from some of the ape species, gorilla, orangutan, gibbon, chimpanzee, to obtain respective precipitates. The amount of precipitate obtained are given below. Based on the given data, the evolutionary closeness of the above apes to humans is. So, the more the amount of precipitate per unit ml, the more closer it is to the humans. So, what they have done is they have injected human serum into a rabbit and raised antibodies. These antibodies they extracted and purified, and a fixed amount of antibody was allowed to react with the fixed amount of albumin from the various ape species. So albumin is the antigen, and human antibody raised in rabbit is the antibody that they have used for reaction with albumin present in ape species, that is gorilla, orangutan, chimpanzee and gibbon, and they obtained precipitates in units per ml. and they have given a tabular column showing how much they have obtained, the amounts of precipitate that they obtained. So the more the amount of precipitate per unit ml, the more closer it is to the human. So chimpanzee is more closer than gorilla, gorilla is more closer than orangutan, orangutan is more closer than gibbon. So as per the options, orangutan greater than gorilla. No. Gibbon greater than orangutan. No. Chimpanzee greater than gorilla. Yes. Chimpanzee has got more precipitate units per ml than gorilla. So it is more closer to humans than gorilla. Gibbon greater than chimpanzee. No. Then next one. Which of the following properties of cellular transport across cell membrane would be applicable for transport of molecules which have a significant hydrophilic moiety but do not undergo active transport? Option A, involvement of special membrane proteins. That is correct. Dependence on osmotic pressure? No. Transport against a concentration gradient? No. Utilization of ATP. Because particles, the cell membrane has got a lipid bilayer. So particles that are lipid soluble can easily diffuse through the cell membrane, as the lipid is the major constituent of the membrane. Whereas hydrophilic substances like glucose do not pass through these biological membranes by simple diffusion. They are transported by facilitated diffusion. That is, a carrier is involved, or carrier mediated diffusion, which take place with the help of transport proteins. So involvement of special membrane proteins is the next one. Which of the following tissues of plant origin have the potential to introduce and transfer genetic variability to their progeny? Eyes of potato tubers? No, because they are vegetative they are involved in vegetative propagation. Then rhizome of ginger, that is also a means of vegetative propagation. Leaf buds of bryophyllum, that is also a vegetative propagation. Seeds of mustard. Seeds of mustard are produced by sexual reproduction. So genetic variation is observed in sexual reproduction only. And these which are involved in vegetative reproduction, they are means of asexual reproduction. So genetic variation will not be observed in these. So answer is option B, seeds of mustard. Next one. During evolution of plants from growth in aquatic habitats to growth on land, which of the following processes would have been important to the plant? Development of mechanisms for vertical growth? No, need not be. Even in land also, even in water also, they have developmental mechanism for vertical growth also. Then development of water conservation strategies. Yes, that they have to, because they are coming from a aquatic habit to a land. So they should develop water conservation strategies. Protection of reproductive cells from desiccation. Yes, that is also one important strategy that they have to develop. Then absorption of synthesized food from the soil. Need not be. So option 2 and 3 are correct. So answer is option C, 2 and 3 only. Next one. If a female who is a carrier of hemophilia gene has a child with a male who does not have hemophilia, which of the following predictions would be correct? So they have told that there is a female parent who is a carrier of hemophilia, and this is the genotype of the female parent who is carrier XXH, and male who does not have hemophilia, so XY, and they get children by fusion of the gametes. So XXH will be the gamete in female and X and Y will be the gamete in male, and upon crossing you will get XX, XY, XXH, XHY. So you get two male children, of which half of the male children will be hemophilic and the half will be non-hemophilic. Similarly, you get two female children, of which half of them will not have any hemophilia and half of them will be just carriers but do not express hemophilic phenotype. So answer is option D. 50% of the sons and none of the daughters will have hemophilia. Next one. Peripheral neurons propagate action potential in response to sensory stimuli. These neurons have different diameters and have different levels of myelination. The diameters and myelination of the different neurons are as follows. Which statement regarding the speed of the action potential propagated by neurons is correct? Now you should know that the velocity of conduction is greater if the diameter of the axon is large or if the axon is myelinated. So as the diameter and myelination is there, then the velocity of conduction is also more. So A alpha neurons conduct fastest while C neurons do not propagate any response. Non-myelinated doesn't mean that they don't propagate any response. Highly myelinated, yes, they and more diameter of the axon. So naturally A alpha will conduct faster, but C cannot be. So option A is not correct. A delta neurons conduct faster than C neurons but slower than A beta neurons. A delta neurons have got higher diameter than C neurons. So and also they are minimally myelinated whereas C is non-myelinated. So A delta neurons conduct faster than C neurons but slower than A beta neurons. A beta neurons have higher diameter than A delta and A beta is more myelinated than A delta. So option B is correct. C neurons conduct fastest while C neurons don't conduct fastest. Then A delta neurons conduct faster than A beta neurons. No, A delta doesn't conduct faster than A beta neurons. So option B is correct. Next one. In plant growth and development, which of the following sequences of events is correct? Sporophyte, mitosis, gametes, meiosis. Gametes, meiosis, gametophyte, sporophyte. Gametophyte, meiosis, zygote, gametes. Zygote will produce sporophyte. The spores will undergo meiosis and form the gametes. So option D is correct. Next one. A person has received enough exposure of X-rays in the whole body to destroy only dividing cells. Which of the following functions would survive best? Cardiac contraction, red blood corpuscle production, hair growth, intestinal absorption of fat. Hair growth and red blood corpuscles are dividing cells, whereas heart muscles are non-dividing. So option A is correct. Next one. Questions 211 to 214. One or more than one of the four options may be correct. Your answer is regarded correct only if you choose all the correct options and no incorrect options. A correct answer will earn 5 marks, a wrong answer or an unattempted question will earn 0 marks. Coming to first question. Neuronal response to stimulus can be probed by electrophysiology measurements and recorded as an action potential generated in response to an input signal. If an experiment corresponding neurons obtained from healthy rat and from hypersensitive rat were probed after an input signal of varying amplitude was administered once every second, the results obtained are shown in the following graphs. This is the amplitude of I and this is the amplitude of this is the amplitude of I and this is the amplitude of action potential. AP is action potential. This is neuron of H and neuron S. So which of the following statement about the experiment is or are correct? Both H and S neuron initiate an action potential when the input signal I is less than or equal to 1. So when the input signal is 1, that is over here, H, and for S is also input signal 1, they don't generate an action potential. See here also there is no record at a time 1 second and here also there is no reading at time 1 second. So both H and S neurons initiate an AP when the input signal is less than or equal to 1 unit is wrong. They don't initiate an action potential. Then both and HS neurons initiate an action potential only when the input signal is greater than 2 units. Yes, when the input signal is, that is here, is greater than 2 or equal to 2. So it is equal to 2 and 3 it is greater than 2. Similarly, in the case of neuron S, equal to 2, greater than 2. Here also equal to 2, here also equal to 2. So what they have told? They initiate an AP only when the input signal is greater than or equal to 2. So at at time 2 second you get an AP, at time 6 second also you get an AP, and at time 10 second also you get an action potential. So at this particular value of I, that is 2, you get an action potential for neuron H. Similarly for neuron S also, at 2 you get, then at 6 you get, and at 10 also you get. But you can see other lines also at 3, 4, 6, 7, 8, 9, 8 and 10 and 11, 12 seconds. That will be discussed in the next option. So both H and S neuron initiate an AP when the input signal is greater than or equal to 2 units. Yes, that is correct. That we already discussed in option B. But only S can sustain it for more than 1 second. This sustaining of the signal by S is shown by at 3, 4, 7, 8, 11, 12 signals in case of this graph for neuron S. So S is able to sustain it, that is sustain the amplitude of action potential for more than 1 second. Okay, so answer is option B and C. Then next, okay, one more option is there. S but not H neurons always initiate an AP when the input signal is less than or equal to 1 unit and is sustained. No, there is no, I told you already that there is no action potential when the input signal is 1. So option D is also not correct. In a plant breeding experiment, scientists crossed two homozygous parental lines P1 and P2 to produce F1 hybrids designated as F1 P1 by P2. Another group of researchers produced a different set of F1 hybrids F1 P3 P4 using homozygous parents P3 and P4. Unfortunately, the seeds of F1 P1 P2 hybrids got mixed with F1 P3 P4 hybrid seeds and with seeds of other parental plants not included in the above crosses. To identify the F1 progeny and their respective parental combination, a DNA fingerprinting experiment was done using molecular markers on 4 confirmed plant lines P1, P2, P3, P4 and 6 randomly selected potential F1 individuals designated as F11, F12, F13, F14, F15, F16. The DNA fingerprint profile of the bands obtained for various plants is given below. So what they have done is, a few of the plant breeders, they crossed P1 and P2 parents and raised F1 hybrids. These parents were homozygous parental lines. Similarly, another group of researchers, they crossed homozygous parents P3 and P4 and raised the F1 hybrids F1 P3 by P4. Now what happened was, the P1, P2 and P3, P4 hybrid seeds got mixed up. So they were not able to know which one was raised by crossing the parents P1 and P2 and which seeds were raised by crossing the parents P3 and P4. So in order to identify, what they did was, they conducted a DNA fingerprinting experiment using the confirmed parental lines, that is P1, P2, P3, P4 and 6 randomly selected potential F1 individuals, that is F11, F12, F13, F14, F15, F16. And based on this profile, they wanted to know they can, or else they can come to know through DNA fingerprinting profile which of the parents' progeny belongs to which hybrid. So this is the, based on the above data, which of the following is or are correct combination of parents and their corresponding F1 progeny. So P1, P2 is equal to F1, 1. So this is P1, this is P2, these are the DNA fingerprinting bands, and this is F11. Now see whether all the bands present in P1 and P2 are present in F1, because F1 must be the cross between P1 and P2. So we will see whether it is correct. Now this band is present in P1 is present in F1 also. Then these two bands present in P2 is present in F1 also. Now these two bands present in P1 is present in F1 also. Then this band present in P2 is present in F11. Then this band present in P2 is present in F11. So P1 by P2 cross will have the seeds F1, 1. Okay, C for whether matching of the bands. Now we'll take B. P1, P2, F1, 5. Now P1 and P2 and F1, 5 is this. This first band present in parent 1 is not present in F1, 5. Then F1, 5 has got a new band over here which is not present in both the parents. Then these two bands present in P2 are not found in F1, 5. Then there is some band which is similar to this band in P1 and P5, but there is no, this band is not present. Similarly here also these are all new bands which are not present in P1 and P2. So F15 will not be the offspring of P1 and P2. So answer B will be wrong. Now take C, P3, P4 with F16. This is P3, this is P4. This is F16. This band is is present in P4. Then this band is absent from F1. Then this band is also absent. This P3 band is present here. Similarly, these are new bands. Then this also is a new band. So P1, P4 cross cannot give you F16 seeds. Now we will see P3, P4 for F14. Now P3, P4 with F14. Now this band is present in P4, so it will be present in offspring. Now this band present in P3 is present in the offspring. This band is also present here. This band from P3 is present here. This band from P3 is present here. This band from P4 is present here. Similarly, this band from P4 is present in this. So the answer is A, P1 to F1, 1 and P3, P4, F1, 4. See how simple the question is. Next one. An experimental setup to study the effect of pH on the activity of the enzyme amylase is shown in the figure. The tubes A and B are made of semi-permeable dialysis tubing. So the membrane for dialysis tubing is semi-permeable. At the beginning of the experiment, equal volume of starch and amylose solution are added to each tube. So into each of these tubes, they have added starch and amylose solution. Now what does amylose do? Amylase will act on the starch, it will digest it and produce maltose. Then buffer solution at pH 3 is added to tube A. So this is tube A where the buffer solution at pH 3 is added, and buffer solution at pH 8 is added in tube B. After 30 minutes, the height of solutions in tube A and B is measured and the solutions in the tubes tested using Benedict's reagent and heating. So Benedict's reagent was used to detect the presence of maltose. Which of the following statements would be correct after 30 minutes of the experiment? So what they have done was they have taken two tubes. These two tubes are made of semi-permeable dialysis tubing, and into each of these tubes they have added starch and amylase solution. Amylase which will act on starch, catalyze it to produce maltose. and the production of maltose was detected using Benedict's reagent. Now another thing is that the buffer solution at pH 3 was added to tube A and buffer solution at pH 8 was added to tube B. So which of the following statements are correct? Now before coming to the explanation, I will tell you that amylase digests starch into maltose. Maltose can be detected by Benedict's reagent, and Benedict's solution will be blue first in the beginning, and as the concentration of the sugar increases, it will turn red. It will turn red especially when higher concentration of sugar is present. Then the optimum activity of alpha amylase was found to be in the pH range of 4.5 to 7. So first one, the level of solution in tube A would be higher than that in tube B due to diffusion. So diffusion cannot happen here because it is a semi-permeable membrane. So it must be osmosis, not diffusion. So it happily eliminate option A. Similar in option B, the level of solution in tube B would be higher than that in tube A due to diffusion. So here also diffusion, so that also will be eliminated. The next one, the level of the solution in tube B would be higher than that in tube A due to osmosis, and Benedict's test would give red-brown color in tube B. Now that is correct. Why? Because tube B is having a pH of 8, and I told you that alpha amylase has got a pH range from 4.5 to 7. So at this pH 8, alpha amylase would digest starch and produce maltose. So the level in solution B would increase than in tube A, and the Benedict's test would give a red-brown color in tube B. So option C is correct. Then next one, the level of solution in tube A would be higher than that in tube B due to osmosis, and Benedict's test would give a blue color in tube A. Benedict's test is actually blue in color, which turns green, then turns red as the concentration of the sugar increases. So I told you that in tube B the reaction has taken place between alpha amylase and starch, and amylase is having an optimum pH for activity at pH 4.5 to 7. So naturally D will be not correct answer, C will be the correct answer. Next one, Huntington's disease is a genetic disorder that leads to defects in the brain function and is always fatal. The affected gene consists of sequence of DNA having multiple repeats of the nucleotide sequence CAG. A factor that determines whether an allele of this gene causes Huntington's disease or not is the number of CAG repeats. The graph on the right shows an age in years at which the patient, each represented by a dot, first developed symptoms of Huntington's disease, and the number of CAG repeats in the allele causing Huntington disease in each patient. From the above graph, which of the following would be correct? There could be factors other than number of CAG repeats that determine the age of onset. Yes, that is correct. Not only CAG repeats, there may be other repeats also that determine the age of onset. Then there is a negative correlation between age of onset and the number of CAG repeats. So as the age is increasing, you can see from the graph that as the age is increasing, the number of CAG repeats are getting reduced. So that shows a negative correlation. That is, as one factor or one variable increases, the other variable decreases. That is negative correlation. So option B is correct. Then Huntington's disease is fatal and is therefore not heritable. It is heritable disease. Huntington disease is heritable and can be passed on to offspring. So option D is also correct. So options A, B, D are correct. Please like, share and subscribe my channel. Thank you.